introductory of molecular mechanics. But before I do so, I just like to wrap up the electronic part. We have developed an effective Hamiltonian that describes electronic coupling of excitations in one of these small rings, and we can now generalize this Hamiltonian to describe actually the whole ensemble of aggregates. It's very trivial because the coupling between these rings is just van der Waals induced dipole coupling, and so it's actually a very trivial extension without any problem, and one can then apply the theory to this whole aggregate, and one can compute the transfer rates and compare them with experimental measurements. And here you see that, for example, if one of these 800 nanometer chlorophylls absorbs the photon within 2 picoseconds, it's transferred to the big ring. The experiment says 0.7 picoseconds, so here you see that one cannot get it exactly right. I think we even understand now a little bit where this comes from. And then you're getting a transfer here in 10 picoseconds between the small rings, and in about 7 picoseconds between the small ring and the big ring, where experiment says it's about half the time. You also get a back transfer because in thermal conditions you always have a forward-backward reaction. Then you see from the big ring you go to the reaction center within 15 picoseconds, 25 observed. You also have a transfer back rate where you get pretty close agreement between experiment and theory, and then you have the first electron transfer here in 3 picoseconds. So you see the overall process can be understood very well within about a factor 2, and so we can really say that we understand the physics of the system. The numbers, by the way, involve the notion that we developed of the partial coherence of the electronic excitations shared coherently, as I said, in these rings. What has not been addressed yet is the role of the carotenoids. The carotenoids absorb at 500 nanometer, and if one develops a theory, one finds that they have actually two functions. So if it's really a wonderful example in actually many electron physics, you can show that the carotenoids can exchange with the chlorophylls two types of excitation: triplet excitation and singlet excitation. So far, everything we talked about was singlet excitations where the spins of the molecules do not participate. But they can also transfer triplet excitations, and that's very important because the chlorophylls, every once in a while, even though they are excited in singlet excitations through sunlight, end up in a triplet excitation. So that they're excited and one of their spins flip and they become triplet. And this is very, very dangerous because they can collide then with atmospheric oxygen. And atmospheric oxygen, when it collides with a triplet molecule, triplet excited molecule, becomes then singlet oxygen. Oxygen is one of the few molecules that is triplet in the ground state, becomes a singlet oxygen, and singlet oxygen is chemically extremely aggressive. And in fact, the modular character of all these building blocks is due to the fact that this triplet excitation catastrophe happens, that the triplet excitation can go over to the singlet to oxygen, and oxygen then eats on the system, and that is why these systems have to be very modular, because you have to constantly repair them, and you don't want to repair everything, you just want to repair what's broken. And so under full illumination condition, every half an hour, every of these proteins need to be replaced. So you see another thing, you have to aggregate, they have to function together, you have to repair them. So the good engineer thinks about all of this. How do I build the bridge? How does the bridge function? How do I repair the bridge? And so here this needs to be taken account. Now, this is such a critical problem that the carotenoids actually have two functions here. When they come so close to they can take triplet excitation from the chlorophylls away, and they can do that because their triplet excitation energy is lower than that of the chlorophylls. So now when the chlorophyll gets excited, it transfers the triplet excitation to the carotenoids, the bad excitation. The good excitation, the singlet excitation, goes from the carotenoid to the chlorophyll because they are excited at 500 nanometer, very high in energy. The energy can go to the carotenoid. But triplets, they have low energy, so they take it from the carotenoids. Now they can of course also collide with oxygen, but it turns out that the carotenoids have such low triplet excitation energy that's lower than that what the oxygen need to be promoted to the singlet state. And so there is not a problem anymore with oxygen. And you see that faithfully, you see the carotenoids being spread over the whole system in order to protect these chromophores against triplet excitations. Now, to do this, the quantum chemistry and many electron physics will tell you that it only works through exchange excitation, not through van der Waals interaction. You must exchange two electrons. Otherwise, you cannot transfer triplet excitation. And so as a result, the molecules have to be completely in contact to transfer triplet excitation. And you see that the carotenoids are either in contact directly with one chlorophyll, or they are indirectly in contact with it. These two chlorophylls are in contact. If this is triplet excited, it can transfer here. This triplet excitation can go here. So you find that in none of these systems there is not a direct contact-to-contact situation between chlorophylls and all the carotenoids in the system, so that they are being protected. So this is just a beautiful outcome of this kind of description. Now the way the triplets, the way the singlet excitation goes to the carotenoids, from the carotenoids to the chlorophylls, is also very elegant physics. Because the carotenoids have actually two excitations that play a role. The first excitation that absorbs 500 nanometer photons is actually a higher excitation in the carotenoids. They have a lower excitation. So the one which absorbs the light is a higher excitation, and the one that is lower excitation is usually after when the photon is absorbed by a carotenoid within 100 femtoseconds or faster. The excitation goes to the lower excitation. This excitation is very good in absorbing photons. This is really bad in absorbing photons because this one is actually, I talked about the low triple excitation of the carotenoids. Now I'm talking again to the physicist. This one is actually a triplet excitation. You can do two triplets, you call that physicist a magnon. Two triplets you can couple to singlet. One triplet up, one triplet down, gives zero singlet. And so this is a triplet excitation, but because it involves spin, it's very poorly coupled to photons, and is also very poorly coupled to the chlorophylls. And this state here is very absorbs very well. Actually, in our eyes, we have carotenoids because this is such a good absorber, such a good light detector, but it is only very short-lived. So it can only couple for a very short time to the carotenoid, to the chlorophylls. Then you go to that long-lived, miserably absorbing, spin-dominated state that is very weakly coupled. And so now the nature has to develop a strategy to get within 100 femtoseconds, which is really short, definitely on the chemistry scale. It is even short on the scale of a physicist dealing, for example, with electronic devices. Within 100 femtoseconds, one has to couple it quickly there. Otherwise, you go down here, and then you have a bad state to couple. So this is a problem you're faced with, and if you look at if you calculate through this, by the way, are highly, in terms of the language of many electron systems that was introduced to us by Todd Martinez a week ago, this is a very highly correlated state. If you want to describe this one, you have to do really well in terms of quantum chemistry. You have to perturbation theories, you can forget it. You have to do really complete CI to calculate that state properly. And this is what you get in a normal carotenoid. You absorb in the second state, you can transfer 29% to these 800 nanometer chlorophylls and another 17% to these 850 nanometer chlorophylls. That together is about 50%, and the other 50% is lost. You go to S1, and S1 is very poorly coupled, only 4% is coupled here, the rest is lost. So many carotenoids, they can get 50% of the energy out of the carotenoid, but the other is lost. But now then, nature is greedy. And if you are getting more light, more energy out of the sunlight, you are having a big advantage in terms of evolution. If you look at the genetics of the system, you find that it is an example for very divergent evolution. It is not like everybody found the same solution and they agree to it, but you find many differences in terms of how these machines are built, because there was incredible competition for sunlight. And now you find that there are many plant species that are modifying these carotenoids. They break the symmetry of these carotenoids in order to get out of this magnetic state better coupling. And what they do is they do two things. They build in heteroatoms, like oxygens and nitrogens, and they build in rings, heterorings. And you can show that these elements are breaking the symmetry of this state so that it mixes with this state, and that then this state has partially the character of this state, and then it's also coupled, and you can then the rest of your 50% you can also get. So you find that those species that get 100% have carotenoids that are not beautiful, like in flamingos. The pink color is a nice, beautiful carotenoid, whereas in plants you see that also in the fall colors they have these very deep red colors and so on. They are often due to these carotenoids that are chemically strongly modified in order to get these 50% not being lost. And so this is really like the same kind of physics that we do in physics for understanding superconductivity, in particular high temperature superconductivity. And so we have to really understand electronic systems very well to understand the intricacies of these systems. It's all nicely described actually in this article, which you also find on our website. And now I would like to go to the rest of the system briefly, namely the BC1 complex and the ATPase. I just would like to show that also these systems can be studied with molecular modeling approaches. And let's quickly go to the BC1 complex. The BC1 complex takes a quinone and takes the 2 electrons of it and the protons, and it uses a trick that I cannot explain to you now, but the trick is that it actually does something really crazy. Rather than turning one hydroquinone into quinone, it takes 2 hydroquinones and turns them into quinones by making 2 electrons over here, 2 protons here, but making on this side a new hydroquinone. So really crazy, but you will see when you look at it, it works well. You take one hydroquinone, turn it into this, a second one, turn it into this, and you took 2 electrons here, and 2 electrons, 2 protons you take putting here to make out of a quinone a hydroquinone. Really crazy. But you know, like it's like how you invest your money. You have to think sometimes very, very oddly to beat the system. And so that's what they do. And as a result, they must send 1 electron down this route to where the cytochrome sits. And they must then send 1 electron here to that other quinone, this way. How do you send 1 electron this way and 1 electron that way? How do you do that? You do that actually quite often. What do you do? take one wire in the kitchen with another wire in the kitchen. No, what do? You throw a switch. Then first one switch goes like this, we throw the switch goes here. And this what this does, you have a domain here with an iron-sulfur cluster that can take the electron, and when the iron-sulfur cluster is here, the electron can go there. If you move that iron-sulfur cluster away, close here, then you cannot, the route is interrupted. So you are moving this domain, and this domain is anchored with a big helix here. It has a single connection, and then you have here this protein domain that can rotate, and it has an iron-sulfur protein that can take an electron, conduct it or not. This was the suggestion. The crystallographers only saw that this domain was disordered under many crystallographic conditions. And they asked then, the crystallographers who solved the structure, they came to us and said, you know, if we could not see if this domain can be rotated. And of course, it was a wonderful opportunity for molecular dynamics. We just took the domain and rotated it around. Here you see that the function of time, the torque we needed, you see going like doop, 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 in several steps. We needed strong torque, weak torque, weak torque to do it. And we found some intermediates that agreed very nicely with the physiology of this molecule. You see, it was a huge simulation that we did already a few years ago with over 200,000 atoms. But you know, it was really simulation that showed that this domain is actually movable, and it is a mechanical switch that diverts an electron either in one direction or the other. Now we come to that wonderful ATPase. And here you see a model of the ATPase. Rather than using words, you can see quickly how it works. Protons go from one side to the membrane, are taken on a ride by a cylinder, and leave them on the other side. And thereby they turn the cylinder into rotation. The protons cannot go straight there and out because there is a blockage, a Coulomb blockage, so they have to go around here in order to make it to the other side. They want to go across the membrane because there are more here than there. And here's a torque that now works itself through the top part of the protein that is held here, that it cannot rotate itself. So the inside axle rotates and changes periodically the protein. The protein on top there has a threefold symmetry, and the torque rotates by 120 degrees, 120 degrees, 120 degrees, and there are now the binding sites here for the ATP, for the ADP and the phosphate. They go inside there. And this is, of course, extremely schematically here, so it's just a basic idea. And so now the binding site is changing periodically. Now it prefers to be an ATP, and then the site likes to be empty, it's been kicked out, and then it becomes a site that likes again to bind phosphate and ADP, and so on. And so by going here, you can show that you can turn mechanical motion into chemical synthesis. You call it rotatory catalysis. By changing the character, the chemical character of the sites periodically, you can make the system rotate, synthesize ATP. And interestingly, it works both ways. If you have too much ATP, it rotates the other way around and pumps protons out. And if you have more protons, you make ATP. It's like you drive your car uphill, you need gasoline, you drive it downhill, your tank gets filled again with almost no loss. So it's not like you need much more ATP to go in the other direction. So it's a very reversible system. It's a remarkable engine. And so first one wants to understand the quantum chemistry. Now I don't have an acknowledgement slide, so I have to just, because I'm too short now, so I have to acknowledge the people as I go. And this is actually the work of Markus Dietrich, he's sitting in the back. And so he studied first quantum chemistry, and he wanted to know what is exactly the chemistry looking like, because it is mechanically modulated chemistry. And so one really has to understand, of course, at an electronic level, the chemistry. And so he built up from the crystallographic structure, that is a structure from Walker in Cambridge, who got the Nobel Prize for having solved it. He took the structure and he, to the best of his ability, solvated it with water and equilibrated things. And then he found a way that was very surprising and new, that actually two water molecules are involved in attacking this phosphate. You see, phosphate is now attacked, and now this is now the transition state. The phosphate now has two bonds, so now this water is attacking it, and at this moment the phosphate is now in the situation where it can now cleave this bond and make then bonds with waters here and turn the system then into ADP. So it's ATP, and here you see the key attack involving two water molecules. And he could show that with these two water molecules, the barrier can be reduced drastically so that in the pocket of the protein, you can actually do the chemistry. And then you can see that if you do the chemistry, you end up with a very high energy level, because the phosphate, here's a product, here's a phosphate, and here's the ADP, the two phosphates and the third phosphate being cleaved. This is doubly negatively charged, this is single negatively charged, and they repel each other, even though counter ions are around that try to dampen that. They still repel each other, and so you have really some kind of like a combustion engine, that you're getting suddenly a situation where you have a lot of compression in the binding site. I don't want to use these words too much because we really don't understand yet what is really going on, but this is what these results indicate, that you are putting it in the binding pocket, you are reacting it, the bond gets cleaved because you have the proper water arrangement, and then you suddenly find yourself in an electrostatic Coulomb repulsion situation with which you can do work. Now, the work you do is to rotate the stalk that you saw earlier, and what you do is you put now the top of this protein on a substrate, you give it ATP, and now you go in the opposite direction, you hydrolyze ATP to ADP, and that makes the stalk rotate, and you can put a big actin filament on it that you can see in a microscope. Here you see this actin filament, and it rotates around when you give ATP. When you stop giving ATP, it stops rotating. And you can also see that you really turn slowly ATP into ADP. You can actually measure exactly how much ATP it's needed, and you find you need 1 ATP for 120-degree rotation. You can measure that quantitatively. You see some further data. You can measure that, and you see that you always step 3 steps. One is here in radians, so it's 2 pi, and so you go 120, 120, 120, 120, 120, 120. And now the scientist who did this, you call it video microscopy for a single ATP, as you can do this. You see that actually the steps are 90, 30. So you often have 90-degree, 30-degree step rather than 120-degree steps. That depends a little bit on the ATP concentration. If you have lots of ATP, you always go 120, 120, 120, but if you are giving a little less, then you can resolve that you actually go 90, 30, 90, 30. And so there you're getting some hints, and now of course this invites molecular dynamics simulation to build the stalk, the protein, top of the protein, put the ATP in there, put it into water, add the ions, and then rotate the stalk. And here you see what happens when you rotate the stalk. You see that you take on the bottom, where the stalk is connected to the cylinder that I showed earlier, you see how you can rotate it. And you can now analyze this. And so here see again how the stalk is made of 2 alpha helices that are actually forming helix. Here you see how, as you apply the torque in time, so here goes time, and here's the length of the stalk. This is the bottom of the stalk, this is the top of the stalk, and you see that the bottom of the stalk rotates very nicely. You apply a constant torque and it rotates, but then you see that at some point in the middle of the stalk, about here, where you rotate the bottom, but the top here doesn't rotate with it. So it works against something. This is a point where you actually interact with the protein, where you turn the torque into work. Now I cannot show you now because of time what you actually do to the binding site, but you can show that they're energetically altered in a way that is very reminiscent of the rotatory catalysis I showed earlier. And now I just want to rather show you what's happening on the bottom of it. On the bottom of it, you saw already in the movie that protons try to go across a membrane, and the only way they can do that is by going on a ride with a cylinder. And here you see the details. You have here a proton conductor, goes from here, can proton being conducted. And now here is an arginine group that carries two positive charges, and the protons cannot continue there. Here's a conductor continuing. We got side groups here that conduct the proton out, this one in, but here's a blockage. And so now, here you have on each of these cylindrical proteins made of 10 identical units. They're in the membrane, completely hydrophobic, except for one group that can, an aspartate group, that can take a proton. And here you see 10 of these groups, one for each of these units. And they can take a proton from this group, rotate around, and give it then to this group, and go on. And this is what you want to resolve in molecular dynamics simulations. And so here we build up this thing. This is actually work by Alec. Is Alec here? Okay, so he's not here. And is Ilya here? Okay, so there are two group members who do this work. And so they built this whole system. And then they first wanted to see if they can actually rotate this ring inside. Is it possible? And they found, yes, it's possible. They apply a torque, and it's actually, you have to apply some reasonable torque, but it's not a really super huge torque. You can really rotate that cylinder inside of the simulated membrane. You can do it with different torques. You can measure then the diffusion constant and the friction constant. And you have then, if you want to model it, you have then these quantities available. And then they wanted to know the interaction. And you know you have this interaction between these groups that donates a proton and takes a proton and the aspartates. And when they let everything loose and started simulations, they actually found that everything broke apart. So here you see that this helix there gets deformed. And when you hold that helix in place, so you say, okay, I don't allow you helix to be deformed, then the blue helix gets deformed. So they form a very strong salt bridge when they transfer the proton, and so somehow it didn't work. And then they found a wonderful way out through a collaboration with Bob Fillingham, who is a professor of biochemistry in University of Wisconsin. And he had some indication that these helices that are outside of this ring are rotating, because he did so-called cross-link experiments, and he found that he could cross-link, let's say, this side group with other helices around here in all kinds of directions, as if this helix is rotating. Totally crazy, he thought. And he came to us, and then we tried to rotate this helix, and you see how we rotated it. You can rotate it straight, but then you don't get a good rotation. It's more, you have to do more like a hula hoop rotation to do that well. And if you do this well, you choose a good kind of rotation axis, like a tilted one, then you can see that you can rotate this helix. Here you see that. And so you have an orrery. You have an outside ring with like ball bearings, and an inside ring also rotating. And so you see how this, we rotate the helix around and around, even by 360, because the loop here is made of dihedral angles, and you know the dihedral potential is periodic. You can rotate and rotate and rotate and rotate the helix. It doesn't wind up or something, because it's a periodic potential here, the famous Ramachandran angles. And so you can really rotate the helix around with rather little torque. And so then he could put it all together in a model. And here you see the model. And you see now, when he permits the helix to rotate, then he found something incredible. He found that this salt bridge here, that the ring cannot continue to rotate, but now you can rotate because you transfer the salt bridge to the next aspartate. And then this helix will rotate. See, at the moment it will rotate. Now it rotates. And now it comes closer to the next aspartate. This is, by the way, the arginine that is so positively charged. And so the arginine gets this group, says proton, you go here, puts it over there, you say you send the proton up here. Gets this proton, you put on there, and pulls it over one by one, and has the protons being loaded on here, and the protons being uploaded on the other side. And so here you have really, apparently, a real ball-bearing kind of rotation inside of the protein. If you would have told anybody that proteins rotate like 10 years ago, people would have said, you're totally crazy, you know, you're in the wrong field, go back to mechanics. But today we know they rotate, and now we see that even they rotate, they have sub-rotations in the big rotation, apparently, if this turns out to be right. And we put it all into a model with the key degrees of freedom. So this is actually, in a way, a very important point in my lecture. And even though it takes now another extra 10 seconds, I like to emphasize it. I think the days are over when you just do a molecular simulation, you look at some little detail, let the protein wiggle, look at it, you know, like this. You have to also have a top-down approach. You have to have an overall model that you link with this simulation. And here we have this kind of model for the overall process on a millisecond scale. It's a stochastic model that describes how everything rotates, how everything functions. The parameters either come from experiment or they come from the modeling simulations. So you come from top down and from bottom up, but if you don't have an overall model that describes the integral function, you're in the wrong department. So you shouldn't do that anymore. You should try to come from both sides and try to link the higher organizational platform with the lower organizational platform, the atomic molecular level with the cellular process level. And that is where the fields are heading. And then you can put it all together. This model is now a stochastic model. You can solve it, and you see here the rotation angle. You see the transfer of the salt bridge and transfer of protons. You put a load on it. This actually rotates against the load that is expected to be needed in order to synthesize ATP on the top floor. So the top floor tries to resist the rotation and thereby using the energy for synthesis. And you see that this whole thing works quite nicely. So this is the end of it. And I hope I didn't end with something too technical, but you know, like, please remember the overall gestalt of my lecture, which was that we had this photosynthetic unit going from the light harvesting step to the electron transfer step, to the step of taking electrons and protons from quinone, shuttling electrons back, proton on the membrane, then understanding how ATPase uses a proton gradient to synthesize ATP, and we try to understand the entire system. We try to understand it, how things work together, but we should also try to understand how the system develops in the first place. How are these proteins genetically controlled and aggregate at the right place, in the right composition, in the right pattern? How do we repair the system? This is a system that is exposed to radiation damage. Usually the cell tries to protect everything from radiation, but here it has to go to the outstation because it gets energy from the sun. It's dangerous, but it has to take the risk because it needs energy. And so it has to repair constantly. How is it repairing itself? These are all the questions that one should try to answer and thereby move on into the biophysics of today and tomorrow, the biophysics of your generation. Thank you very much for your attention. So there could be now a few questions, and we of course also have some general questions about the meeting, so we might want to keep those questions short, that we can still have some general questions that you might have. Yes?
I was just wondering if you had some of those movies, particularly the notation one, the software chapter available as supplementary material or something on your website?
Yes, we have some material on the website. So if you go there, you find it. You can also send me, some of it is just at the moment being reviewed, and so it might be officially available any moment. So you can get much of it. Some of it is already reviewed and some of it is being reviewed. The ATPase, the BC1 is already all out. So BC1, light harvesting, reaction center, electron transfer, so they're all published. And the ATPase work is more recent. Okay.
How to add force for the single helix rotation?
Excuse me?
For the single helix rotation, how did you add force?
For the single
helix.
head. Oh, I see. Yeah, so we designated, that was done by Barry. Oh, I didn't mention him. So Barry, you saw him many times around. So he did the work on rotating the stalk. And Barry designated, if I remember, 8 atoms on the base of the stalk. And then he applied torque to them so that you basically rotate them around. You have to, get the geometry right. So he just basically had to do his cosines and sines right to be sure that you rotate the 8 atoms on a perfect circle.
I believe the question was about the helix rotation.
Oh, the helix rotation. Oh, the helix rotation was much more involved. So we first rotate the helix around the fixed axis, or helix, let's say like this, but we rotate it like this. So let's say, I don't want to show you my belly, but no, I do. So I go like this, and then, the belly is really sweeping too many lipids along. And so it was possible, but it was not very graceful. It looked like a little stupid. And then we realized that each helix has a certain reptation cylinder. And so then we realized we can rotate it in this reptation cylinder. You saw how it rotates. It's hard to describe, but it rotates. This rotates here, this rotates here, this rotates here, this rotates here, this rotates here. It's not a perfect rotation, but you still, basically, you're moving the sides of the helix around and around. And that is, and then we found, if you do this, then, so we gave the system more flexibility in rotating. We didn't say you have to rotate around this axis. Then we found that you actually can rotate the helix very easily with very little torque.
Can you elaborate a little bit on the stochastic model, how you determine the parameters?
So yeah, there was mainly the issue is mainly what are the key degrees of freedom. So we identified then there are basically all angles. There's a rotation angle of the entire cylinder. Then there is a rotation angle of each of these helices, and we had to basically designate, like you have a 10 helices, but only 2 are interacting at each time with a proton channel. And so, but we took an inner model 4, because whenever to tilt, you have a periodicity, then this one moves into the volume, one out, or if you rotate back. So we always have each time 4 of these angles also included in the model. But we have 5 degrees of freedom in the model. And then we assume that all the interaction, to do it simple, is in a plane. So it's not quite right. Some of the groups are outside, but we just assume, let's assume it's all in the plane. And then, however, we took all the geometry of the groups as we know it. For example, if some aspartate moves to the arginine, then we know the distances. Or for each of these groups, we also assumed the center of mass for the charge. So then when it moved, then we really calculate the geometry in order to calculate what energies do they have at each moment in time. So we basically did, to the best of our abilities, in a reduced motion space with reduced degrees of freedom, we made a geometrical energetic picture of the system. And then you have certain parameters in your, you can formulate it either as a stochastic differential equation or as a Fokker-Planck equation with the same kind of parameters. One is more in probability space, the other one is more in position space. And we used the stochastic equation because it was more feasible mathematically. And then you have a certain number of parameters, and it turned out that the parameters we could either get from experiment, or when we needed, we got them from the simulation. So there was no parameter, was just pulled out of the air. And then we put them into the model. And then you have basically coupled stochastic differential equations. And that's nothing really very particular. And you can actually turn them into coupled partial differential equations for the probability distributions. And so they are well understood kind of equations then.
Do you have the time rates right? Or?
Yes, the time got right, but that partially is also due to some numbers from that agrees with the experiment. But some of them, like the friction for the cylinder, was our number that could have slowed it down tremendously if it would be off or made it too easy to rotate, and that turned out to be a pretty good friction constant. But sort of this is a kind of modeling that you need to apply. You really have to be an expert in kinetic theory and stochastic differential equation or Fokker-Planck equation. That's basically the kind of equation you get in non-equilibrium statistical mechanics to do this model. So if you want to know, for me, what is the best field you can invest in if you go beyond the normal one? You should know classical mechanics, electromagnetism, should know quantum mechanics and equilibrium statistical mechanics. But if you then say, what is the next field I need to know to be good in theoretical biophysics, then it is, in my eyes, clearly non-equilibrium statistical mechanics of classical systems, but also to some degree of quantum systems. Actually, I didn't understand your question. Can you maybe make it as short as possible, because then I have a chance.
You have this example, you applied rotation artificially. Do you know, are you thinking about making an example where you have protein which self-rotates, not with an applied force, but by itself and by some properties of the system?
Okay. So yeah, so the problem is that that we have a huge time scale gap. The function is millisecond. The cylinder moves in the millisecond. The microscopic process of proton transfer and local injection are just really like picosecond to nanosecond processes. And so you have this huge gap in time scale. Those we can simulate, but the long simulation, the long step we cannot. And so the only way we can link to the millisecond process is that we guess the key degrees of freedom involved in coupling the proton transfer processes to the rotation. So there's a big leap of phase. We have to, but like without thinking, there's no science. Yeah, so if it would be all, you just put it in the computer, it goes, and the computer publishes your paper, fine, but that's not the world in which we live. We have to think. And so here we have to think. We have a huge time scale gap. We have to think about what could be the key degrees of freedom. And of course, I only give you the success story. I cannot tell you the many. This was actually a really difficult project that ate basically the lives of two postdocs. They worked on it for many years, and there are many false leads. So you try this, you try that, and after a while, you understand the system better. You get also feedback from your errors, and you think you know what the degrees of freedom are, very often, and this was true here too, the key breakthrough comes actually from experiment. In this case, we had some beautiful data, actually were also nicely analyzed by Tick, who sits in the back for the Fillingham group, about the cross-linking studies that gave us a clear pattern for the rotational degrees of freedom explored by these helices. So I think that's very clear that they must rotate. And the two people actually independently found this and published this, and I think there are few people who would not believe that anymore. And so that was important. Then we could put it into the picture. And so we really have here pretty simple elements that we know that the ring rotates pretty much from the video microscopy. We know that the helices rotate from cross-linking studies. The structures are not so good because they are mainly NMR structures that we had to piece together from different substructures. There are some element of uncertainty. Yeah, and then we basically thought these other degrees of freedom, and we tried them out and they worked. So if you want to do it tomorrow, you will find yourself as much information as possible on the overall process. You build your model, you do simulation, then you ask what degrees of freedom are important. Usually it's a problem of a coupling. You want to couple one process, a very microscopic process, to a larger scale, longer time scale problem. And you have to find out what degrees of freedom achieve that coupling. And then you try them out one by one. So it could well be that we just tried the wrong degrees of freedom and people will laugh about it. Maybe we make history by showing what crazy things people do. But it could also be that I think we have a good chance that people will say in 10 years, my God, they figured it out. They saw not everything precisely, but they saw some of the key elements of this important machine. Okay.
So it looks like there's sort of intermediates in that there's sort of a quinone transfer step and then sort of I guess, a hydrogen transfer step on the side from C. Is that just diffusion or is there some sort of guide to that so you don't lose quinone molecules?
So there's a big pool of quinone molecules usually, and hydroquinones. They're just swimming around, they're going through the membrane. So that's at least the picture people have, but that is one of the things we want to visit by putting everything really into one membrane, see how the quinones really migrate from the reaction center to the BC1 complex. But let me just tell you that this, once you become an engineer, you become more real, not just a pure scientist. You brush with the real world. So here we have this ring. We have the reaction center inside, and it binds the quinone, turns them hydroquinone, puts them out. But how do they get through the ring? Now, it turns out that in some species, probably they have to make it through the ring. And one of the things we want to do is to see if we can pull the hydroquinone inside and outside of the ring. But there are some other species, and that was a gene I almost started to tell you the stories, but then I decided I didn't have the time, that codes for one of these elements of the 16 rings that is actually a cap, an end cap. So the 16 ring then is a ring, and then suddenly the end cap. So it has an opening. Why? Because it would be great to have an opening, because then the quinone can go in and out quickly. You call these elements PUF-X. You saw the gene name was PUF, and the name of the gene, because you know what it was doing, was X, so we call the PUF-X gene, and we saw now that's actually the first crystallographic structure too, that there are end caps, and so the ring is actually not completely closed, the big ring, so that's at least in some species, and so that the quinone hydroquinone can shuttle in out faster. Likely it is that in some species that's not the way it is, but in many species definitely there is actually a door. So, like once you ask, how does this thing get in and out? You realize, oh, it doesn't get through this ring. There must be some opening. There must be a chemical modification of that ring. And so on and so on. You also see that the BC1 complex is a pair, and it has a very clear valley in which these quinones go in and out. So you also see that they're not randomly going through, but there's a nice door, come in, here we process you, and here's reception, and there's an exit door and a parking place and so on. So, okay, then I would suggest, I was thinking, what could I do for a summary? I think, you know, like, I mean, I like to say that it was a lot of work for us and for my coworkers, and I like to use this moment to thank them all very much for their work, and I will do so more when we meet with them later today. But I like to also thank you, because you were really, and it comes really from the bottom of my heart, and I know this, I'm speaking also for all of us who have organized this summer school. We really enjoyed very much all of you, and it was really for us a nice experience. We felt all the time that we had some people who were responding, who were interested in what we prepared for you, and who really grew through the 2 weeks and learned something, and that was for us a great pleasure. And I think the best way of actually summarizing is that I would just say, let's go through these wonderful pictures that Mu took and look at them and enjoy them together. So I just go here quickly to, and so here we have the photo gallery. Oh. So I think we start out with yesterday, where we had the wonderful beauty contest, and so I think all the presentations were shown here. They were truly technically an achievement for such a short time. You really got a lot done, and so you will recognize your presentations here. And this guy was funny. I really enjoyed his humor, and except that he tried to bribe the instrument. That was really a little bit low. So you see here the timing, then you being measured, and so yeah, that's a fellow. Is this a lady? Okay, so that's a cave. That's a cave, and so more of the cave. Okay. And here, the cluster building, I was actually really enjoyed coming up there and seeing you guys turn into hardware gurus. You probably never did this kind of thing before, and but you see that's all just normal things. It's not something you can do as well. So, okay, so you recognize that. This one is now some older pictures from the cafeteria, and I think we went through some of them already. And so I think this recalled the whole week. This was the haptic device, and our people who gave you hopefully good advice. Oops, oh, no, I did. Where would I now? Here, if I go here? No, where would I just? Oh, good idea, yes, of course. Oh, great. Okay, no. No? Well, maybe what I do is I... What?
Web server's being a bit slow.
Do you think so?
Yes.
No, I think there's something... I think what I do is I go quickly back into... I just go...
It's having a signal.
What is?
The web server.
I start
We'll get there.
refresh. No. Okay, so you can look at those pictures yourself later. Yeah, so otherwise I'm a little empty-handed because I had to do the 3 lectures today that took my time, but I also felt, you know, it was just a very nice event, and, you know, like it doesn't require much of a philosophical statement now to say that we actually learned a lot. We had a good time with you. That was just wonderful, and I hope you got something out of it, but many of you told us that already, so we are happy about it. And I hope that we feel that we will stay in contact in some way or other. BioCore is a nice way of doing that. And so that's all very great.
Speaking of BioCore, I've had a few people ask me, and you will not lose your BioCore account once the summer school is over. You will keep that account so you can continue using BioCore. You will remain in the 3 summer school projects so you can come back and review everything that was done in there at later time if you want to. And you're more than welcome to use it for your own projects if you wish to do so. If you have questions, just log on and ask us and we'll be able to help you out.
So for us, this was not a routine event, and so we feel like we got to know you during the 2 weeks, and we will always remember our first summer school, and that means that you are always welcome. We really feel like you are our friends now, and if you want to visit us sometime, you are invited, or if you want to collaborate, you know that we are running a resource for NIH where we are really supposed to collaborate with people, not only writing papers with them, but also providing services, helping them with our programs. Sometimes we can also give some people computer time on our clusters. We are not a computer center, we don't want to compete with NCSA, but, you know, once in a while we can give also some computer time. And so we would be delighted to keep in touch with you. And if any of you as a student looks for postdoc or post looks for faculty position, now you have seen how awful it is here in Urbana-Champaign, and you may nevertheless want to consider working here. It's a place where, you know, like, maybe you laugh about it, but, you know, you can work very well. You know, it's I mean, for me to go home just takes 5 minutes, and every life is easy in. It works again. Life is easy in here, and if you really want to get some exciting science done, this is actually not a bad place. So let me see that we continue then. Oh. Let me see where we were. I think we were about here. Oh, okay, good. So we see the pretty ladies again. So, okay, so this was, okay, so here, this one we didn't see yet. Okay, so hope you recognize yourself. And there will be a highlight on our website describing the summer school starting right after the school. So, okay, so it was really nice this year. I've really enjoyed seeing you all working there and doing things by yourself, and learning by doing. That's really important. So here we have these cluster sessions. So that was good, yes. Okay, and here we of course have the grand finale. I just doesn't fit on my picture, on my thing here. Can I make it smaller? Or maybe we just scan through you guys here. Oops. So I was, remember that. Will we get a high resolution picture?
That's highest.
This is already high?
Yeah.
Ah, okay, very good. Okay, so we can we can recognize, because you have to really look carefully to remember people. And so we don't provide this photo because you have it now available on the web, and you can download it and make it into frame.
I will also provide the full size of the picture so that you can print
How, so we didn't understand you quite. Can you repeat?
Some people ask if I can provide the full size, the larger size of file so that we can print. I'll do that after I organize all the pictures that I took during summer school and link that on the photo gallery. And also I have a brief announcement. So we would like to take a picture for everyone who volunteered to write a diary. So I still have 3 people that I still haven't got a chance to take a picture. They are Han Liang, Craig, Jolie, and Walter Ash. Please come to me after this session and I will take pictures.
Did you get this? Are these 3 people here? You and you, and so if you could come, that he takes your picture, and we have your diary entry with your photograph. It's getting more and more difficult. Okay, Rosemary?
I don't know if it's already been said, but if you were supposed to submit a diary entry and you haven't done so already, please do so today. We have, I think, only 4 or 5 out of the 10 at this point.
So please don't leave town without it. Okay.
We'll beat you up.
No, we don't. Okay, so that was it, and now we can have some last questions that you may have at this moment.
I think someone else already asked this at the beginning of the summer school, but I just like to make sure. I thought you said you would provide the participants with a CD.
Yes.
Exactly would be that in that CD now that the summer school is coming to the end, and you probably know that. Do you know when it will be ready?
Okay, so
It's going to be copies of VMD and NAND to actually use, and copies of tutorials. As far as I know, the thing should be ready not today.
what do you have?
It's just t-shirts, there's nothing on their video. Why don't you tell them and send them the box of the protein?
Okay.
Just tell them this is 3%.
Okay.
Show them the model of the protein and tell them.
Okay.
There's 2 medium ones for the girl and the boy.
Okay.
I went to all stores in town.
Oh, shit, I should have given you more money. Okay. So, so are there further questions? Good, then the only thing left is that we had yesterday the beauty contest, and the main prize will be that we are making you small models like this of your protein, but much smaller, but like about quarter of the size, and we have to send them to you, and we couldn't get them in time because the printer takes 10 hours for one protein. So we will try to make the proteins that you worked on and send them to you. And so since you might be disappointed that there was nothing else, then Elizabeth just went out and got you this beautiful shirts with the insignia of our university. And so 2 medium size for the ladies. And for one smaller guy. So she sized you. And so if you have a question which size you think you are, Elizabeth knows it all. Better than mother. Okay, good. So they are here, and maybe you in a nice way you are handing them to our winners. And okay, good. Then we have another afternoon session, and I wish you all a safe trip back. And I'd like to say to my wonderful colleagues who helped here so much with it, that I will thank you separately. But I like to, say already that it was really great what you did. It was just absolutely unbelievable how you really did on your own so many good things for the school. And as you know, we celebrate tonight a little, and I owe you a lot. Okay, so have a good trip back.